to have a missed class meeting. So this recording is as important as if we met class today. I obviously have a cold and or something like that, and I'm not, going to come in because it's a fresh cold, and I feel okay, not too great. But anyway, the lab class is not meeting today. I posted that on both Bright Spaces for the lecture class this morning and the lab class this afternoon. And so you do not need to send me notes on this missed lecture because I missed it. You're getting used to the idea that we have to participate in every class meeting. If you miss, you have to take pictures of the notes that you take within a week to show me that you're participating. So again, take notes on this. It's as important as any of our other class meetings. This stuff will be on the test and so forth. And we just need to try to keep rolling forward because we're poised for these quizzes next week. So probably the first thing is, pre-med club. So we talked about that on Tuesday some. Then, I want to answer questions. So if you have questions about this class meeting, bring them to class, write them down, right? Got to write them down. You won't remember them. So, write them down, bring those questions to class. You're looking at chapter 1, and we won't do a concept chat next week because we'll have the quizzes at the end of class, so we won't actually get the concept chat started. They're very straightforward, and we'll just do them every class when we don't have something else going on. So we walked through all these on Tuesday. Here's the list of stuff you should get going. It's the same as if I had been in class today. It's all laid out on the Bright Space, the quizzes, and so make sure you watch all of this lecture today, before next week, because it's going to fit in, and we're not going to wait and study it before the exam. It's fitting in now in our course, and we're moving on from this material on the start of class on Tuesday. Then also, you should watch all the videos. That's the more important assignment. If we look at what to do for this week, the most important things are work on getting a book, then next read the syllabus. There's a video of it as well. It's just 10 minutes long, so make sure you understand all the rules of our course. Then the next priority is to watch those videos. Watch all the videos before next Tuesday. We'll have a quiz on the first 3 on Tuesday. At the end of class, we'll have a quiz on the next 4 and into class on Thursday. But watch them all before Tuesday, because on Tuesday we're going to have a class assignment that we'll do in class. And so try to make it to class if you can. Remember, if you miss class, you have to send me notes. If you miss class, there are no makeups for the missed assignment or the missed quiz that we'll have on Tuesday. For example, everything like that is announced ahead of time, and those assignments are they're not worth a ton of points, and it's a motivation to get to class. But also, if your car breaks down, if you're sick, like I am, so you don't have to report that to me. We'll just keep rolling forward like that. Yep. Okay. So keep up with Bright Space stuff. Watch our syllabus. Be square on that. If you don't have the book quite yet, I first put the first 2 chapters on electronic reserve, so read the first chapter. What's going to happen is the 7th chapter is something that we're going to jump into with the videos. So the quiz videos are moved toward chapter 7, point toward chapter 7. So I'll start talking about chapter 1 this week, but I won't finish it. It's an overview of these fields, but also it has a lot of specific details that we'll need to know for the exam. So highest priority is watch this lecture, then watch the videos for all the quizzes next week. It's I don't know how long. It's not a lot of, the videos are each 10 minutes long, so it's not a lot of trouble. And you need to know the material well. We'll go over it in class. The quiz will be end of class, so bring your questions to class and bring lots of questions. But then start reading chapter 1. And again, I'll talk about it some in this lecture, but then we really won't get back to it much next week. Maybe some, but we'll be kind of leaning into chapter 7. All right. So I described on Tuesday that an informal subtitle for this course would be Molecular Mechanisms of Cancer. And I described why that's a good thing for us to focus on. And it fits into this, that when we're talking about cell biology, we're talking about molecular biology, biochemistry, genetics, genomics, those are all tied into our course. We're standing next to that table and picking examples from the table. And so it's challenging. So let's pick a topic that you haven't had a course on yet, cancer biology, and it's a good one because of how it fits into our course. For example, our author is a world famous cancer biologist, and he studied the nuts and bolts of cancer, and this course is a nuts and bolts course, and it's a hard course. I talked about the first time I caught it, you should have lots of questions because you should anticipate that you can understand everything in this course. Everyone can understand everything in this course. We just go through a lot of material, and it's your job to say this material is stuff that I'm confused on, whether it's a video, whether it's a class meeting that you follow up on, or in class, or especially in our book. Yeah. The best way to do that learning is to be writing it down. That's why we're going to do an assignment next week, is to get in the habit of writing stuff down and drawing it out. All this stuff is very physical, and cause and effect relationships. This protein binds to that DNA all the time, and matter of fact, a lot of common sense stuff. And so, you can learn this stuff, but you got to be active. You got to write it down, draw it out, and say it out loud to yourself. I say flashcards are a good tool in this kind of course because they're going to be short answer questions like, what's this? What's that? How's that work? How's that work? And also that writing that you're doing is you're preparing your study guide. Students often ask, is there going to be a study guide for the example? Everything I say is the study guide. That's why we have class. And I know that's kind of a snappy answer, but yeah, I've distilled it down as much as I can. So it's your job to be working on learning it and also working on preparing, stuff that you can review for the exam. It's not going to be helpful if you just go back and try to read the book again or, like that, right? So draw it out, write it down. We'll do that in class on Tuesday. You should just be getting in the habit of doing that, being an active learner. And that's why I make these videos, to look like that, to make them more accessible for that active learning. If you have some clay and construction paper, get that out. It's memorable if you do that. Appreciate that there are different components in this, so it could be multiple drawings. These are just the DNA strands, and the parts that are DNA are really important for us to appreciate that that's DNA, and it's not a transcription factor. Transcription factors are proteins. So we're going to talk more about this, but we actually are leaning into Chapter 7 when we talked about FOXO, and also further on into the third chapter of our course, Chapter 17. And that's because, I got to get my little drawing pad turned on here. That's because FOXO needs to go into the nucleus to do its job. FOXO is a transcription factor. And that's what we're learning about in Chapter 7. That's what the second set of videos are all about for our quiz next Thursday. In those videos, we're talking about muscles. Here we're talking about any cell in an animal and rate of aging. In muscles, this transcription factor would be MyoD or myogenin. Both of those are in those clay model videos. So MyoD and myogenin just kind of get made and do their job. This one is regulated. This one is regulated by phosphorylation and exclusion from the nucleus. It's not in the nucleus. It can't do its job. Okay? So it's really important for us to appreciate that. That's coming out of there. And so we have this big picture story, and it's going to be a list of transcription factors and how those get turned on and off. And the first example is myogenin gets turned on by being made. When it gets made, it does its job. FOXO, our next example, is it gets made, maybe it gets to do its job. If there's a growth factor present, FOXO doesn't get to do its job. It's turned off. How's it turned off? Primarily by phosphorylation. We'll see other examples of proteins that get turned off or on by phosphorylation. So that's an important one, and we'll add proteins to that. They get phosphorylated and they get regulated. FOXO is also excluded from the nucleus. We'll see another example of that in our course. So we want to have two examples of regulation of FOXO. It gets phosphorylated, turned off, it gets bound and excluded from the nucleus. Here's an image from chapter 2. And we're not going to read chapter 2 in our course. Chapter 2 introduces some chemistry that is prerequisite type chemistry primarily, and some biological chemistry. So here's just a chemical reaction from chapter 2 to show what's going on with FOXO. Here's FOXO, the substrate, and here's FOXO, the product. And what happens is an enzyme phosphorylates the FOXO protein, and we need to know that that enzyme is always called a kinase. A kinase is an enzyme that catalyzes the addition of phosphate to a target molecule. In this case, it's a protein, and it's a very specific enzyme that phosphorylates that target molecule. Sometimes these kinases have multiple targets, but they're very specific targets. As a result, we have about 600 different kinases in our human body because they turn a variety of proteins on and off. And so we want to know a lot about kinases in our course. And the other substrate the kinase uses is ATP. And that's something you know a lot about, and it's kind of curious to see here. Why is ATP involved? And the answer is that this substrate has two forms. And I'm just going to, instead of drawing it in a purple shape, I'm going to draw it like this, where I'm going to draw it like that. Okay. And then it's going to be like, and then it will fold up, we'll say, into more of a thing like that. So it could be more open or closed. So it has two shapes, two conformations. And both of them are stable, and it could potentially flop back and forth. But it turns out this conformation is more stable, so it just never flops into that conformation, rarely, like that. This is very much similar to the analogy of if you have a light switch for the lights on the wall, and you're going to turn the lights on and off. The switch isn't floppy. The switch is not just physically flopping back and forth, and the protein needs to be ready to do that, but not so similar in their energy states that these two conformations would be flopping back and forth. There has to be some kind of energy barrier here so that it stays in this conformation. And then once it flops over to this conformation, it stays in that conformation. And it is an energy barrier. There's an energy barrier here. You have to apply some energy to flip that over. It's not floppy. And so that's the reason why kinases are so cool, because they use ATP. And we're used to knowing that ATP is an energy intermediate. And what is it doing here? What is that energy using here? And the answer, the energy is using to actually flop this over. Yes, the protein gets phosphorylated as part of the process. But a key concept is the phosphorylated protein is in a different conformation. And so our textbook artist shows this figure. And see how the textbook artist has really emphasized that 1433 protein is binding. Ooh, we really jumped off track there. That 1433 protein really appears to be binding to the phosphates. It should show more of it binding to FOXO. FOXO has changed shape, and the artist is flawed in not representing that. And it's satisfying to see that FOXO is like a teeter-totter, but there's like a heavy kid at one end. And so that heavy kid is going to stay down without some energy put into the system to flop it over. And once it's flopped over, now the heavy kid's up in the air and stays up in the air because there's a bigger weight that got put on the other side. It's very much like that, very much a physical story. Yeah. So good example of a transcription factor getting regulated by phosphorylation, and then the change of the shape of the protein in combination. The 1433 is also probably binding to the phosphate groups, but that's probably not enough information to bind to, right? Because it's like, it's not just any phosphate group, it's on FOXO. So there must be some binding to the surface of FOXO. And it's too bad that the artist and editors didn't catch that in that drawing. But that happens kind of a lot in science textbook. Okay, so we're working on that list, and we'll just keep revisiting it because we want to know more examples of how transcription vectors get switched on and off, because they switch on and off gene expression. Okay, and so that's that, and then we'll move on. We'll start trying to get rid of some of these. Why isn't that erase? It should erase. I don't know why that one didn't erase. Well, we might be distracted by that for this lecture because it should erase with that button. Let's see. Oh, okay. So now we're going to talk a little bit about Chapter 1. And so in Chapter 1, there are 3 sections. Let's just look at them and what's expected of them. So it's kind of curious that the authors, a little bit curious, that the authors start our book with the origin of cells. And the reason it's curious is because not very much is known, and it's a tremendous mystery. It's clear that all life on Earth is related because there's so much similarity at the biochemical level of all life on Earth. I'll show you in a minute. Maybe we'll just scroll to that now, and I might take a minute to find that. But I'll just show you this. When I mentioned before on Tuesday that this course is an intro bio course. And yeah, this is a little bit far of a scroll for this. Maybe I didn't even include it, but I think I did. Sorry. Yeah. So this is a handout from when I teach intro bio. And I just try to say there are 7 themes. We're going to try to cover 7 things. And just to mention them here, because this is cell biology, and I mentioned on Tuesday that intro bio course is introductory cell biology, introductory microbiology. So let's just look at this course, this list. So all life is composed of cells. That's called the cell theory, and there are 2 parts to it. The cell theory emerged in the 1850s, where people realized, no, life doesn't appear spontaneously. Life comes from pre-existing cells. And so that's the cell theory. And when we look at those cells, they're astonishingly similar. All cells exist in a water environment. That's number 3. All cells are bounded by a membrane, and in all cases, it's primarily a phospholipid molecule barrier. It has proteins and other kinds of molecules in it, but in every cell on Earth, there are a lot of phospholipids that make this lipid bilayer. So every cell on Earth has the same strategy to have the outer boundary of the cell be a membrane composed like that. All cells inside of them have really similar chemistries. There is DNA, that's the next one on my list for intro bio, number 6. DNA is the primary heritable molecule of all cells on Earth, and DNA's big job is to code for proteins. So DNA will learn here codes for RNA, and then RNA codes for proteins, and that's true for every cell on Earth. And so it's also true that those genes are regulated, and the cell has all the genes it's needed. So those are the big concepts. And now I have to scroll all the way back to wherever we were to say that the authors start out wanting to talk about the origin of life because that's when we would have gotten a common story of the origin of life. But it's still a big mystery. So we don't want to put too much time into this section because our course is so focused on that there is a lot more to be discovered in molecular and cellular biology, but there aren't a lot of great big mysteries. There are mysteries, yes, and I'm not saying that there's more work. There's great big discoveries that will be made and so forth, but it's different than talking about the origin of life, where you're mostly just going, yeah, we don't know. Yeah. So we move on quickly to the second and third sections. There are 3 sections in Chapter 1. And the second section talks about the different organisms. In this research, all the learning that we're going to do in this course, the research is done almost invariably, and yeah, I don't know why that's going to keep popping up. It's a little bit defect, but I'll just write this word here. These organisms are called model organisms. There's a set of organisms that we want to talk about, introduce, because they are the source of the bulk of the work that got done to make the discoveries that we need to learn about in our course. And so you're familiar with the idea that a fruit fly is a model organism, and there are some other ones. I mentioned this little nematode that Cynthia Kenyon studied the rate of aging in. That's another model organism. There's an organism called a zebrafish that's sold in every pet store, and that's a model organism. Mice, of course, are model organisms, and there are more. There is a model organism bacteria called E. coli, and there's a virus that infects that was the original model organism, because molecular biologists are reductionist scientists. They want to go to the simpler system, so they started out with this little virus that infects this bacteria. And so the book is going to want to introduce those, and we'll talk about those. I'm going to go straight to the third section. The third section says it's about tools of cell biology. So remember, I said on Tuesday our course is cell biology and molecular biology. Well, we're going to study cell biology, especially in section 3. And of course, there's tremendous overlap between molecular biology and cell biology, but there are some distinct features and principles. And so it's interesting that the book devotes a full section on it, but it's primarily because the book goes on to really emphasize molecular biology. There's lots and lots of molecular biology in our textbook, in our course. So it's good to introduce these here. This is cell biology, and we'll use some of these concepts, so we want to pick them up and take them with us. And so here's some examples of things to learn, and I'm just going to talk about these two right now. So one thing is there's a lot of microscopy that gets done by scientists in the course we're going to talk about, and so let's talk about it a little bit. So we're going to go into an example of cell biology, and that's a big part of our course, but not the one that we're going to have quizzes on Tuesday, and not the one that is really kind of the bulk of most of our course. So cell biology really started out, it started out before the 1950s, but people usually started saying cell biology in the 1950s because scientists looked at electron microscopes. And so we want to say that first, that there was a long history, the cell theory in the 1800s. That's cell biology, right? But the 1950s is when scientists really said, this is a field of cell biology, and it's because they looked at stuff under a microscope, and they looked at real high magnification images of eukaryotic cells under a microscope, really kind of for the first time in the 1950s. So that's what we think, is we got to find out what all these little organelles do. They didn't know. Some had been named, they'd seen them under microscopes, but they didn't have a clue. So let's find out what they do. And so we want to just talk about cell biology in a way that introduces some of the principles by describing some of the work. Okay, so the first work, they used an electron microscope, and the type of electron microscope they're using here is called a transmission electron microscope, a TEM. And so you could be asked about an electron microscope in a 2-point question on our first exam or the final, because the final exam covers our whole course. And the question could be like, what is a transmission electron microscope used for? Or it could say, what are 2 types of electron microscopes and what are they used for? Because we're going to look at a second type in another minute here. And so a transmission electron microscope means it sends electrons right through the sample. The sample has to be a really thin slice to view, and that's what's done here. This wasn't a whole cell, it was a slice. They had done like slicing a loaf of bread in these little sections, and so they're looking at a thin section to allow the electrons to go through, and the electrons go through, and so we see a image through the cell or the tissue like that. Yep. So here's a different kind of electron microscopy just to compare and contrast them. This is called scanning electron microscopy, and scanning electron microscopy is used to look at the surface of a thing like a bug. And so to do scanning electron microscopy, what's done is the surface of some kind of material a lower magnification material is coated like with gold or some metal. And so what scanning electron microscopes do is they look at the electrons bouncing off that surface and they see what really looks like a 3D image. These are generally lower magnification. These are higher. And scientists use transmission electron microscopy to look into a cell like this and look at the details at high magnification, or they look at little tiny things like a little tiny virus at high magnification, because this magnification, of course, is famously a lot higher in resolution than light microscopes. This scanning electron microscope, and there are other kinds of electron microscopes, is kind of famous, and it's different because it's not used for high magnification. It's very low magnification, but it's used to look to see great deals in great detail about the surface of an organism that would be too small to take a picture of, right? So, you could look at that under a light microscope, but that wouldn't look very interesting under a light microscope because the light microscope has a transmission. Light microscope has to send light through the sample. So, two types of electron microscopy, two different applications, two different ways that they work. Yep. And so they said, let's start figuring these out. So the next thing we're going to do is, I don't know if I have this numbered, but let's just look at these numbers. So we're going to talk about electron microscopy, and we just talked about it, so we can ask about it on the exam. The book talks about multiple different types of light microscopy, and like a confocal microscopy. And in the same way, we just tackled electron microscopy, and we said, let's boil it down. What's the details here? You should be ready to answer questions on the exam of like, well, what are the big types of light microscope that are mentioned here, like confocal microscope and super resolution microscope, and a 2-point question. Well, what is that? What's it used for? Is it light microscopy or is it electron microscopy? And then a little highlight of, well, what's it used for, right? Like that would be it, right? So we want to get more learning out of chapter 1 than what I'll talk about. I'm welcome to ask questions. Some of these will come up during our course, but we want to have a little intro to them right now. So the next thing I'm going to talk about is two things that are famous in cell biology, and one is homogenization, and that's where we're going to take a cell and we're going to break it up, and then we're going to separate things. So let's get the mitochondria, let's get the nucleus, let's get the lysosomes, and we're going to purify them. And the way they're purified is by centrifuging them, by putting them in a test tube and spinning hard. All that does is the centrifuge spins and increases the force of gravity. So when we spin in a centrifuge, we can talk about the speed, the revolutions per minute, how fast is it spinning, but we can also talk about something called the G-force, and the G-force is the force of gravity. And so the force of gravity normally for us is 1G, and if we spin something, the G-force, and it's just the same as gravity, it's just pulling harder, it's higher, so we could go up to 5G and so forth. So when we spin really hard, little tiny things start to come down. They start to sink or precipitate to the bottom, and we could make a pellet or other things. So we want to talk about that, because what they're going to do is they're going to grind up the cells, and then they're going to spin that cellular extract to separate these organelles and get purified examples of different subcellular organelles. And that was a famous activity they started in the 1950s and learned a lot about the purified components. So identification of lysosomes with differential centrifugation, identification of peroxisomes with differential centrifugation, like that. So lots of that kind of work beginning here and getting a lot pure. So all these different things could be purified by breaking them up and centrifuging them. So let's talk about one example, and that is we're going to take cells, we're going to break them up, and we're going to centrifuge them in a liquid that allows us to make a pellet and a remaining liquid. And the remaining liquid is called a supernatant. So we have a pellet and a supernatant. And things that are big and have a high density will go to the bottom. And so what we're going to do is make an extract, and we're going to make the nucleus go to the bottom. The rest of the cell contents will be up here. So ribosomes, endoplasmic reticulum, and the cell bits of the cell membrane and so forth, all up there. And we can throw the supernatant away and study the pellet. So we're going to make a nucleus, a nuclear pellet like that, and we can study that. Okay. So the way we're going to do that is we're going to homogenize. So homogenize means to physically break the cells up. And we're going to use this thing that's called a Downs homogenizer. I don't think your book mentions it, but we just learned about it. And a Downs homogenizer looks like this. It's basically a test tube that's got an opening here and an opening space here so it can hold a bigger volume. So you can fill it up with your cell extract like that. And then this plunger is going to go inside. And that plunger ball has a specific diameter. And the diameter has been very carefully constructed to be a little bit smaller than the inside walls of the test tube. So that when you're plunging up and down with the Downs homogenizer, there's a little gap between that ball and the wall of the test tube. Okay? And so you're just sliding it up and down here in that distance. You don't go all the way to the bottom. You just go up and down, and there's a little reservoir here to hold that liquid that gets displaced as you go up and down. And you just do that 3 or 4 times, and it's just a gentle thing. And what this gap is the gap is too small for the cell to fit, but bigger than the nucleus. So what happens is that physically, and there's a little tiny bit of a detergent in here and other components, molecules to help with the process, but it's really a physical breaking up of the cell because it can't make it between the wall and the head of the homogenizer, and we're going to break it up. So we could have some different size diameters to try to help isolate other things, but this is a really famous one. Okay, so and then we take that extract and we spin it in a centrifuge, and we'll have a pellet, and that pellet will be intact nuclei. Let's think about this. We could take these cells, right, and we could use the Downs homogenizer to break them up, and then we could spin them. And the nucleus would be in the pellet. So in the absence of growth factor, where would FOXO be? And the answer is most of the FOXO would be here. That's where FOXO wants to be. It wants to get there and do its job in the nucleus. So you can see how cell biology is used in combination with molecular biology. FOXO is really a molecular biology story, and we can learn about its physical location in the cell. On the other hand, if there was a growth factor present, and you can just imagine setting up an experiment, right, where you have 2 conditions, and you say, where's FOXO? Well, one way to test where FOXO is to simply homogenize the cells and ask, is it in the pellet? So on this one, it would be in the pellet. In this one, it would be in that supernatant. Okay? So that's just an example of using centrifugation to start to separate components of the cell. And so that's talked about some in chapter 1, and it talks about some different kinds of centrifugation. So that one we gave was just easy. The nucleus is big and has high gravity. You can spin things really hard, and it's still always just that G-force notion, but the G-force can be tremendous. So you can actually get really surprising things happen. For example, we're used to thinking of something like sodium chloride in a solution. And if you think about it, the sodium chloride is the same molarity throughout because it's just floating around. Well, we can switch to a different salt, and it's going to be cesium chloride. And we'll look at the periodic table. Cesium chloride is a much bigger atom than sodium, and it's dense. It has a high density. So if we spin that salty solution of cesium chloride really hard, we actually can get a gradient of its concentration. with higher gradients down here and lower gradients up here. So higher gradients down near the bottom. It's not pelleting. We're not spinning it that hard and long, but we're spinning it so long that we start to get a gradient of concentration, or gradient molarity, and that also means a gradient of density. So when we start to spin in cesium chloride really hard here, and we use what's called an ultracentrifuge, we start to get a gradient, and we'll see that instead of pelleting, we can have some samples start to float at a specific gravity, because there's a range of gravity in that test tube like that. There used to be these big floor-standing ultracentrifuges. I want to just show you this. Here are the scientists is holding this big heavy roller. It probably weighs 20 pounds, and they're going to use big volumes to do these experiments. Here the person is holding the centrifuge rotor, the part that spins, and the test tubes would be up here, and the test tubes are smaller than your pinky. And this centrifuge is an ultracentrifuge that just sits on a table. It's the size of a toaster. And there are little ribs here, and it blows a stream of air with such intensity that this thing spins 100,000 times every minute. So, ultracentrifuges range in their size and capacity for doing great big experiments. Now, let's just take a quick look at this. So you can see that the density of some of these biological materials ranges. Not by very much. It's very similar to water, which has a density of 1, but it's a little heavier than water. Yeah. And a little denser than water. Yeah. So more dense items would sink. All right. And so we're going to see that. And and that includes things like DNA itself. Yeah. But these molecules are so essentially relatively so small that it really takes a long time to spin them to have that kind of effect. But here's an experiment of a type of ultracentrifugation that we can learn about, and it's a special kind called equilibrium density gradient centrifugation. And you don't need to necessarily be able to list all those words. It'd be great if you could. But that's the full name of this type of centrifugation, and it's that type where we're going to set up a cesium chloride concentration gradient. The way it was set up initially is the person marked this on here, where to set the tube up, they already helped set up the gradient because they put in a couple milliliters of a solution of cesium chloride that already had that concentration, and so a certain molarity and a certain density, right? Because cesium chloride is really dense. And so they made these 3 stacks by just gently adding them on top of each other. So 3 separate stacks. And then when they spin it, they've already set up somewhat of a gradient, and it gets us to become a smoother gradient. And so they put their solution in, and they can just have their sample mixed throughout, and then they spin it. And what happens is that these biological materials will purify in a little hazy cloud. So this is a type of virus called a bacteriophage that we'll talk some about in our course, because it was, I mentioned just earlier today here, that it was a really prominent model organism early in molecular biology history. And so here they're purifying phage particles. What they're purifying from was these were some E. coli bacteria that were infected, and they were broken open, and the viruses that were being assembled inside were purified. And the scientists can collect that by piercing the tube. The tube will be made of plastic typically, and they could pierce it with a needle and actually suck that out, get that sample out. And and so that's just showing that they're getting the purified sample out. So that's a type of ultracentrifugation that's famous because it was able to purify different things. Like, here are the ribosomes floating at this concentration and this density. Yep. So here's just that indication that cesium is quite a bit heavier than sodium atom, and that creates a situation where you can actually spin really hard and get this salt concentration gradient and a corresponding density gradient set up in a tube. And that lets you not just make pellets, but it also lets you have materials float at specific densities. Yep. Here's a transmission electron micrograph, so type of electron microscopy of a little tiny bacteriophage. Those are the ones that were just purified, and you can see they're purified. So there are other debris that were present of E. coli, and maybe some unbroken E. coli in the initial preparation. And by spinning in the ultracentrifuge, we've separated these little phage away from everything else, and we have a pure mix of these phage, and we can study them. We can study the proteins in the head, or inside the head. There's a little stretch of DNA for each of the little bacteriophage. And again, that's what this was. This purified material was looked at under an electron microscope, and it was quite well purified bacteriophage. So just a little taste of what that part of Chapter 1 is trying to cover, is to try to introduce some of those principles. And the way that the scientists in cell biology and molecular biology like to do that is they like to explain a method, like an experimental method. And so we'll do more of that. In every biology textbook, the authors try to put in some methods to try to help the students understand and encourage them to think about the scientific work. We'll do more of that. So our author does it a lot. We'll try to describe methods and not just facts, but, well, how do they know those facts? Because there are reasons to say why we'll do that, and it has to do with just the whole approach, and we'll talk about that also as we get started in our course. So the other part of this course is molecular biology, and our book really doesn't introduce that in the first chapter. One of the reasons is because there's a third or fourth chapter that's called Fundamentals of Molecular Biology. So, it wants to introduce a lot of that because it's a bigger part of our course. And we're not going to read that chapter for a while, but we are introducing molecular biology before we jump into transcription regulation, Chapter 7, next week. So here's an example. Back in the 1940s and 50s, and it continues to this day, scientists studied this little virus, and it's called a bacteriophage, and it infects E. coli. And that's showing it, landing, binding to it, and infecting the cell by injecting its DNA. And you can see that even though it's just a simple virus, this virus has a really complicated structure. Those are all proteins that are the covering of the virus and the function of the virus in its infection process. Really simple. There's no brain going on here or anything like that, right? The virus isn't even alive. It's just a structure of proteins and DNA that can infect this cell. And so scientists started picking on this. And so that's part of the first chapter, but we won't mention it again. But we need to just have a little bit of understanding of it as a model organism, and the original model organism for molecular biologists to study. For example, in the 1940s, early molecular biologists, before Watson and Crick discovered the structure of DNA, early molecular biologists added a radioactive label to the protein and a radioactive label to the DNA. And the radioactive labels were different. And then they said, which radioactive label goes into the cell? And the answer is the DNA goes into the cell. That's the first evidence that DNA is the heritable molecule. That's the 1940s. And so that's, deep origin of this field of research. And that led to the focus of Watson and Crick on DNA and so on and so forth. So there's a little bit of deep history here. Again, this virus is still studied a lot and studied for the, kind of fundamental principles like transcription. It was used to study transcription at an early time. I put these in because these are the same concepts, but they're just more advanced. Instead of picking on that little virus and trying to teach you the principles of molecular biology with that little virus, I'd say, let's go to the cell that you're familiar with. It's a cell that's going to become a muscle in our human body. Yeah? So I don't know why I put that in right there. Oh, I guess this is a little bit out of order for what I was thinking about. And this just shows that in Scarborough at Maine Health Institute for Research, we have students. So these are just examples of 2 undergrad students from several years ago who worked in a lab there. That's the lab leader. So Professor Michaela Reagan, and they are in Scarborough, and we have students and former students that are in those labs down there, and I'll talk about that in class because it's a research opportunity that is about cell biology, molecular biology, and it comes up each school year. So the applications will come up in about a month or so for next school year. And so here we go on back to molecular biology. So Jeff Cooper is a molecular biologist. Historically, that he was a young guy in the 1970s when people were really first getting started on this. And he studied a protein that's called RAS. And the RAS protein is mutated in about a third of human cancers. And it is called an oncogene. It's a normal gene that's present in our body, and it's doing a normal job. But when it gets mutated, that can help contribute to causing cancer and uncontrolled cell growth of cancer in our bodies. So that was his original work. He started working on it in Wisconsin, and then he moved to Boston. And when he first worked on it, and of course, no one knew what any of this stuff, right? But here again, and this is in chapter 16, that's the third chapter we'll talk about. Here in chapter 16, and we looked at this idea with FOXO. We had that FOXO picture where FOXO's in a cell and it could be activated or inactivated. This is a different example from chapter 16, and this example we'll learn in great detail when we get to that chapter. And what's going on here is we're looking at a cell, and we're looking at the location of RAS. RAS is inside, basically inside of every one of our cells, and a related gene inside of every one of every animal cell. So there's going to be a signal outside the cell, some kind of hormone or molecule, and we'll learn about those. And it's going to bind to a receptor. The receptor is going to get activated, and it's going to lead to a pathway. And this receptor doesn't lead to FOXO, leads to a different transcription factor that's down here, and unfortunately has to be shown in a separate slide. So the target that we're going to want to talk about is a protein that is a transcription factor that gets phosphorylated by this long series of proteins getting phosphorylated, and it's called ALK. So we'll add ALK to our list of examples of MyoD and FOXO, and we'll add ALK to the example of FOXO and ALK have to get phosphorylated. In the case of OXO, to turn it off. In the case of ALK, to turn it on. So what's the role of RAS? The role of RAS is in some kind of growth signal pathway. So a growth factor is going to stimulate growth. It's going to do it by activating ALK, and it's going to do it by activating RAS on that pathway. Okay? And so that's what our author studies, and we're going to learn a lot about that. And so a part of introducing molecular biology is to say, let's put it in context. So we could study RAS, and where's RAS at, right? We could do a Downs homogenizer and separate it. We find RAS is always inside the cell membrane, and it's stuck to the cell membrane. This actually shows that RAS is covalently bonded to a little phospholipid that anchors it in the inner side of the cell membrane. And it floats side to side really fast, so it's showing, well, it's not really showing that, but it diffuses side to side, but it's always there because its function is right here at the cell surface where inside the cell there's going to be this response. We'll learn a lot more about it, but just to show you a little bit about where we're heading. Yep. So in doing that, we're introducing molecular biology, and molecular biology is very physical. That's one reason why I wanted to start with that cell biology example of, well, where do you think FOXO is? And we'll be like, it's either in the nucleus or it's not in the cytoplasm. And we can prove it even though we can't see it. So it's a physical story. And then Molecular biology is reductionism, and it's like we're just going to break this system down. They started out with a really simple virus, that little bacteriophage, and they said we're just going to disassemble it to its parts, and we're going to study each of its parts, and we'll find out what it does. So that's a real dominant strategy in molecular biology, and to do that work and get it done, they pick on model organisms in labs, and so those are concepts that are part of molecular biology too. Lots of talk in our book about now that virus, not so much anymore. E. coli, the bacteria, not so much anymore. More and more of it's talking about, like we did, where we started our course with a cell we like to talk about, muscle cell. But nevertheless, still, model organisms, still a really reductionistic strategy, and then done in labs, controlled environments, and just, experiment. Setting up more and more experiments like that. So here's an example of our model organism that gets talked about in the first chapter, and it is a fish, and it's called a zebrafish. And it's studied because it's a vertebrate, and it is, so it's fairly closely related to us, but it has external fertilization. The mom and dad fish just release sperm and egg, and fertilization takes place outside of the organism. That's very different than mammals. Mammals are hard to study because we have internal fertilization. So here the zebrafish is popular because the embryo is outside the organism. And then another detail is that it has yolk, and the yolk is clear. So even when the embryo is early and is in the middle of all that yolk, you can still make videos of it. And here they're making videos of it, and they're using a protein from a jellyfish to tag the nervous system. And the nervous system is glowing green. This little fish is living. It's normal. It's happy. And I already mentioned in the clay model muscle differentiation videos that scientists can make a mouse glow. It has this protein. This kind of organism has been genetically modified. And in a test tube, the scientists built a special gene, and the gene included parts of both the jellyfish and the zebrafish DNA, and they call that a transgene. And this organism that's been genetically modified is called a transgenic organism. The transgene has been permanently inserted into the DNA and the chromosomes of this zebrafish, and we have a zebrafish strain or line that is, it could be totally normal, wild type animal, but it just has this extra transgene in it that causes the cells to glow. The DNA does not glow. It's the protein product that glows. So we'll learn a lot more about that, primarily because we want to understand the organization of genes really well. And one way to understand the organization of the gene well is to break them up, physically break them up, put them back together, and stuff like that. So we use this example a lot in this course. I won't talk about that right here, but maybe I should right here. So people will say, wow, why do they study fruit flies? Why do they study mice? And there are reasons, like they grow fast, they grow well in lab, they grow the same each time, they reproduce at a young age, so you can do lots of work with them. But there's a bottom line reason why we would study a model organism, and that's because it's closely related. And the closely related features of it are all about the genes. And so I already mentioned that you could find RAS in all animals, all multicellular animals. You can find FOXO in all multicellular animals. What am I saying? I'm saying that, where'd that go? That there's a concept called ortholog, and that is that, let's say we pick a gene from a fly and a gene from mice, and they're really, really similar, and they're doing the same function. Our interpretation is that gene was present in the last common ancestor. We're used to thinking about the organisms, that's saying that fruit flies and mice are related because there was an ancestor, like maybe a fossil, that was the ancestor of both those. Here we've dropped down to the gene level. Here's a modern gene in a fly, and here's a modern gene in a mouse, And by studying the gene's function and the gene structure, we say they're really, really the same thing. And the only way to explain that is they must have had a common ancestral gene. And those are called orthologs. And so we want to know this definition very carefully and clearly. There are two genes, and they're in different species that are understood to share a common ancestral gene. And I bring that up now because we want to bring it up again and again and again, and say that in this case, that's the reason that we study model organisms, is because they have orthologs. These are all these are secondary reasons. The number one reason we study a model organism of any kind is because it has orthologs of other genes that we're interested in. The last thing on our list here is something that's very common in our course, is that discoveries get turned into new tools. And that this is a perfect example of that. A scientist discovered that there was a jellyfish that lives in Puget Sound outside of Seattle, and that jellyfish had a gene that coded for a protein that's called GFP, for green fluorescent protein, and that green fluorescent protein caused the jellyfish to glow. When they discovered that, another scientist realized, well, I could take the DNA for the GFP gene and put that under the control of a zebrafish gene and put that into a zebrafish. And here are the scientists that did that. This is the scientist that did the original work on the jellyfish. This is the scientist that put the gene into a transgene and a transgenic source. In his case, he studied the nematodes. This scientist's contribution was they found a variety of mutations of the GFP gene that glow different colors, because it was very valuable for scientists to have a cell glow green and a different cell glow blue and a different cell glow red. And in fact, in the clay model muscle differentiation videos that I made that we're going to have a quiz on Thursday, the mice, there's a mouse that I show that says this mouse has muscles that glow red. It's a transgenic mouse, and it has muscles that glow red and other tissues glow other colors because it's had multiple transgenes put into it. It's a transgenic mouse, and early on in the embryo, after it's been fertilized, it has all the transgene, but none of the cells are glowing because only the protein glows. So after a while, the cell starts to make muscles, and we start to see red glow, and so forth. Yep. So these scientists actually won a Nobel Prize about 15 years ago for their work, and their work was taking a discovery of nature, just the diversity of life on Earth and all the cool things about it, like jellyfish glow, and turning it into a tool. And that's really common in this field. In molecular biology especially, it's driven forward by discoveries being used to make powerful tools. And we'll talk about why that glowing cells with GFP are powerful tools. Yeah. Okay. So just examples of, putting them in different species and seeing them glowing. Originally from that jellyfish, there's the protein, and that's that. All right, so I already jumped ahead and talked about this today, so we're doing great on that because I've talked some about Chapter 1 stuff. And in Chapter 1, there's quite a bit of cell biology. The authors introduced these concepts here. We can answer questions about them in class. I want to show you that these are important ideas. I consider this to be kind of at the introductory level of intro bio, which is a prerequisite. So when we run into something like that, you're like, I know what the endosymbiotic theory is from intro bio. Well, you want to refresh your memory of it here and write it down and make flashcards over these types of stuff. Okay? Yep. Then talks about in the second section of Chapter 1 I mentioned, they talk about some different model organisms and features that make them valuable to study, like that. Okay, so a couple of concepts about that I want to show you is one is I like to show my intro bio students this figure, and maybe I'll stop with this figure today. And so it looks like this, and what it's showing is On this scale on the left it shows in meters and micrometers the sizes of different things. So, here's a person up here in a couple meters like that, and then we drop down to smaller and smaller things, right? And it's on that scale. And down at this, and I seem to have lost my pen function. Oh, there it is. Down here, we start to get down to this level that is, below the visible level. And here, below the visible level, like that, and we see cells. And our whole course starts here and goes down all the way to atoms. And so I like to try to draw a comparison. And so what I tell my students is, imagine that a cell, and kind of an average human cell, was actually the Boston metropolitan area. And, some of you have a lot of familiarity with Boston. So when you say, we're out here in Concord, right? It's an outer suburb of Boston. So 30 miles across. A lot of people, a lot of stuff going on. Imagine that's a cell. Now, if you're not familiar with Boston, don't worry about it. It's just a big city that's about 30 miles across. And, I'm using, English units here because in the United States, people have still more familiarity with that. So if this were 30 miles across and this were a cell, how big would some of these things be? And the answer is, if we drop down to, say, the atom, and we'll look at a water molecule. So there's a water molecule. A water molecule would be about the size of a basketball. And it's physical. It's a real thing. It's a physical thing. And we want to appreciate that scale some. And we'll come back to this some and be able to rank things based on size and get a feel for the significance of that with regard to size. So we go up from there, and you see that, a ribosome is the size of a house. And there are lots of ribosomes. Anytime we talk about a molecule in a cell, there are lots of them. There are tens of thousands of ribosomes in every cell. Similarly, there are tens of thousands of houses here. Yeah. One of the defects of this analogy is we're used to thinking of Boston as flat. The cell is actually a sphere. So, we have a big sphere. So there are lots and lots and lots of houses. And then we get up to this level. Also, just down from this level are almost all the proteins, and they range a lot in size. But if this were Boston, the ribosomes would be about as big as a house, and then the protein products would be a little bit smaller than that. We'd have things like trucks, big trucks, and little cars for big proteins, little proteins like that. And then up bigger than that, the smallest bacteria would be, starting to be up on the order of like a neighborhood. And mitochondria would be more like, almost not a suburb, but a bigger area. Maybe a bacteria would be like a mall, and mitochondria would be like a neighborhood, and we're getting up to that then full-size cell. And we want to appreciate that because this is happening in context, and we're combining both that molecular biology with cell biology to really understand things. And so we'll try to appreciate these physical sizes and their relative sizes and how they matter in the function of the different stories we tell. Okay, so let me stop there. We'll pick this up from here on Tuesday. So again, I'm not going to summarize, what you got to get done because I talked about that at the start of the course. Okay, great. Sorry, I'm sick. I'm sick, and we'll see you on Tuesday. All right.